Hey guys.
Hi, Casella.
Hi.
Tess wants to say hi.
All
Yay.
right. I am trying, for some reason, I have Derek's screen being shared right now. I want my screen. Okay, can you guys see my screen? Anybody? There we go. All right, how about now? Can we see it? I don't see anybody's. Let's see.
Yeah, we see it.
Okay, thank you. All right. There we go. Thank you. I'm going to move my chat screen. So if we can start with chapter 9, that way I can kind of go through and answer your questions as we go. I'm going to close my door really quick. Okay. Any questions on chapter 9? Nothing. Cell cycle, regulation. Okay, compare and contrast cyclins and kinases and growth factors. So we'll start there. Let me find that slide. Somewhere. Okay. So cyclins and kinases are both regulatory proteins, which means they're on the inside, and they can kind of act as the brake in the cell cycle, where growth factors stimulate other cells to divide. So those are more like the gas, and a lot of times those are external signals. So that's coming from another cell that then stimulates a cell nearby to start dividing. So you can think of it growth factor, right? We need more cells to enable growth. So this PDGF, platelet-derived growth factor, stimulates the division of fibroblast cells. Let me see. So hopefully that works. They all help control the cell cycle, meaning depending on what factors or what signals that the cells are getting, that either promotes cell growth and promotes the cell going through the cell cycle and dividing, or inhibits it. All right, explain the evidence, okay, for cytoplasmic signals using figure 914. I believe that's right before this. Yes, okay, so here's figure 914. So in this experiment, they took two cells and put them together. So one cell was in, for example, in this experiment one cell was in the S phase, one cell was in G1. The results were, when they put these two cells and fused them together, that the cells entered the S phase. So whatever phase, actually we'll talk about experiment 2 first. So same thing, we have a cell in the M phase, and then we have a cell in G1. When they put these two cells together, the one in G1 entered the M phase. So it entered the M phase even without chromosome duplication. So molecules present in the cytoplasm tend to, I'm trying to think of the right word, like spur the cell cycle on to the next phase. So the cell will move on to whatever phase the farther cell is in. I don't know if that's making any sense. So for example, here we have one cell is in S, one cell is in G1. So both cells then are going to be in S because that's the further stage in the cell cycle. Same thing here. One is in G1, one is in M. So then the cell is going to immediately enter M. So conclusion here is molecules present in the cytoplasm control the progression of the cell cycle. Okay, then we have, explain the relationship between chromosomes, sister chromatids, centromeres, and kinetochores. My son is texting me wondering where the spinach is. Explain the relationship between chromosomes, sister chromatids, centromeres, and kinetochores, including how many are part of a replicated chromosome pair. Okay, so I don't think I have a total slide for this, but I can kind of show you on the beginning picture here. Maybe. Okay. So, great. So each one of these, so these would be the sister chromatids, right? So each one of these is actually a chromosome, but when it's duplicated and when they're together, we call them sister chromatids. So chromosome, right? Chromosome, but when they're together, we call each one a sister chromatid until they're pulled apart. Once they're apart, we refer to them as chromosomes again. Centromeres, so centromere is just the part kind of in the middle of each sister chromatid that is kind of pinched inward. So you can see this diagram says sister chromatids. So this is the centromere. Okay. The kinetochore is just a region on the centromere. So you can see here, these are the kinetochores. So this is the part of the centromere that actually binds to these spindle fibers and helps these spindle fibers attach. Okay, so how many are part of each replicated chromosome pair? So this right here is a replicated chromosome pair. So if we're talking about one pair of chromosomes, we have two sister chromatids. Each sister chromatid has a centromere and a kinetochore. So right here, this would be one chromosome pair, replicated pair, two sister chromatids, two centromeres, and two kinetochores. So each sister chromatid has its own kinetochore and own centromere and attaches to its own spindle fiber, right? Because then these shorten and then the sister chromatids are pulled apart. Okay, can you do a brief rundown of the different stages of a cell cycle? Sure. Let's see where that is. Let me go back here. Okay. So a cell cycle is made up of mostly interphase, right? You guys saw that when you were looking at the cells, the onion root tip cells. So most of them are in interphase, which is the preparation for the M phase, or they're non-dividing. So they're in G0. So again, interphase is about 90% of the cell cycle. G1 is when the cell grows, right? It has to grow to prepare room. S phase, synthesis, so DNA is made there. G2 is when organelles are made, right? We have to duplicate all the organelles because we're going to have two cells. We need double the amount of organelles. Centrosomes, which are, I typically refer to those as centrioles, so we don't get confused. Those are duplicated, right? That's where those spindle fibers come from. And then, so G1, SG2, all part of interphase, which is this whole part right here, right? 90% of the cell cycle. And then this, the M phase, key thing to remember about the M phase. This is both, you think M phase, mitosis. Yeah, it's mitosis, but it's also cytokinesis. So don't forget that part. Mitosis, remember, is division of the nucleus. Cytokinesis is division of the cytoplasm. So at this point, cytokinesis is when we end up with two cells. Before then, we just have two nuclei that are still in one cell. And then mitosis, we have our PP map. Any other questions from chapter 9? I'm not seeing anything else. Done with chapter 9? Moving on to chapter 16. All right, questions on chapter 16 then? Oh, okay. Back to chapter 9. Compare the amount of DNA in each part of the cell cycle. So, for example, if I could give you any amount, right, of DNA. We could be talking about milligrams, for example. If I weighed the amount of DNA, or if I said there's, I'll just throw out a random number. Okay, let's say there's 10 milligrams of DNA. That's still way too much, but 10 milligrams of DNA in G1. In S phase, there would be 20 grams, right, of DNA. So I have to double the DNA. So remember, DNA is tightly wound into chromosomes, right? So those chromosomes or the chromatin, which is kind of the loose jumble, gets condensed into chromosomes so that it's easier to copy. So those and be pulled apart. So the DNA just gets duplicated as well here. So let's say we have 10 micrograms or milligrams of DNA here. We would have 20 here, still 20 as we enter mitosis, and then as we move into cytokinesis and end up with 2 cells, then those 2 cells would have 10 milligrams of DNA again. So it's just kind of getting you to think of DNA is tightly wound into chromosomes. Okay, anchorage dependence. That is, I believe, towards the end. Or is that in chapter 16? Can somebody remind me? Anchorage dependence, is that in chapter 9 or chapter 16? I get confused because it talks about 9.3.
Yeah, 9. It's in 9.
Okay, thanks. Oh, here we go. Okay. So typically, and this kind of has to do with cancer cells, because typically what happens is that cells stop dividing at certain points. So one of the things that I'm just going to talk about, density-dependent inhibition as well. So crowded cells stop dividing, right? Have you ever wondered, like you get a cut on your finger and your cut heals, but you don't end up with this huge bump on your finger? Somehow your finger knows when it has enough cells and then it stops dividing, right? That's density-dependent inhibition. So when cells touch each other, then they don't send signals anymore for new cells to be made and divide. So those typically are external signals. Anchorage dependence mean that typically cells have to be attached to something in order to divide. So again, that's an external signal. So basically all that means is cells require a surface for division. Cells aren't going to divide and make more cells if they're not resting on something, if there's not a layer underneath. So cells divide to fill a gap and then they stop. So you can see here in this Petri dish, it's just showing you that anchorage dependence, the cells require a surface for division. But this does not happen in cancer cells. So cancer cells do not need a layer in order to keep dividing. Okay, ready for 16 now? Maybe? All right. Questions from chapter 16. Okay, is there going to be, okay, so we're back to chapter 9. That's okay. Is there going to be a question on the test where you're going to ask us how many sister chromatids are in G1, like what we were saying in class? Yes. So you do need to know again. So for example, again, let's say here, if I say, well, again, we'll just stick with 10. Okay, if I have 10 chromosomes in G1, I have 0 sister chromatids. right? Because those 10 chromosomes have not duplicated yet. In the S phase, then those chromosomes do duplicate. So then I'm going to have 20 sister chromatids, 20 sister chromatids, 20 sister chromatids until they get pulled apart, right? And then here we're back to no sister chromatids, just chromosomes. And then remember, for each sister chromatid, we have the same amount. So however many sister chromatids we have. So here we had 20, and we would have 20 centromeres and 20 kinetochores. Okay, describe the two main sources of developmental information for the early embryo. Okay. So I believe, I don't know if I got rid of that slide. So one of them is cytoplasmic determinants. So this is just different stuff in the egg cell, so RNA, proteins, other substances, and they're distributed unevenly. So those influence early development. If we look at the picture here, it just shows, here are all these cytoplasmic determinants, and you can see they're unevenly spaced. So when fertilization happens, when the sperm fuses with the egg, and this now zygote, right, when the sperm fertilizes the egg, it becomes a zygote. Then when that zygote starts to divide, right, it becomes 2 cells, then 4 cells, and then keeps dividing. Those cytoplasmic determinants get divided unequally, and that is going to impact the development. So that's one major way. The other one is signals from other cells. So induction is where signal molecules from embryonic cells cause changes in target cells. So interactions between cells, basically, and those cytoplasmic determinants that were in the egg to begin with. Explain how inherited predisposition and other factors can contribute to cancer. Okay. Oh no, I just somehow closed my little chat window. Okay, there we go. Let's go to the end here. So we kind of talked about this in class, that there's lots of different ways to develop cancer. I'm looking for this slide. So 6 changes have to happen. So you can have a genetic or inherited predisposition. So you could have a mutation in a gene that is passed on from parents to offspring, but that is just one of the factors, right? So 6 factors. It showed here you'd have to have loss of tumor suppressor genes, activation of a RAS oncogene, maybe loss of another tumor suppressor gene. Sometimes they think some cancers can be influenced by different viruses, but again, we need typically 6 different things to occur in order for cancer to actually happen. All right, differentiation between multipotent, pluripotent, and totipotent stem cells. Let's see, that is back up here. Somewhere. Okay, so stem cell, different types of stem cells. So they can actually, they're unspecialized, so they don't have a job yet. They can reproduce itself indefinitely, and then they also can give rise to a certain line, depending on what type of stem cell they are. So 3 main types, totipotent, are able to differentiate into all cell types, including placental cells. So these are stem cells that are isolated only from early embryos. So at the blastocyst stage, which means first few cell divisions. So very, very early on is the only time when we ever see totipotent cells. Embryonic stem cells are actually pluripotent. So those are capable of differentiating into all cell types that make up the body. The only reason these are different, pluripotent than totipotent, these can't differentiate into the placental cells. That's the only difference. Okay, so typically, again, for a lot of research, they're interested in using embryonic stem cells because those are pluripotent, meaning they could create any cell. So goal here is to repair damaged or diseased organs that typically cannot regenerate. So nerves, heart cells, brain cells, things like that. And then totipotent, no, we already talked about totipotent, multipotent. These are typically adult stem cells. So for example, like bone marrow cells, those cells can give rise to all types of blood cells. So white blood cells, all the different types of white blood cells, all the different types of red blood cells. So they're much more specialized. There is as well the induced pluripotent stem cells. They've actually created these in the laboratory. So they are adult stem cells that can be reprogrammed to act like embryonic cells. So that's a huge advancement. Again, there's always ethical issues with using embryonic stem cells. So these are an option for some researchers, and depending on different countries and the different laws that are in place. And just know that, well, I guess it depends on if we get there. So we'll leave it at that for now. Okay, describe the effects of interference with cell signaling pathways. Okay, so what can happen? Man, I keep exiting out of there on accident. Okay, what can happen with cell signaling pathways? So that kind of gets into this. So if there is interference with the pathway, then it doesn't work correctly. So for example, here, if there's a mutation in this RAS protein, instead of needing a growth factor to come in and bind, right, this growth factor acts as a ligand. This is kind of going through that whole cell signaling process again. Only when this ligand binds does this whole cell signaling pathway happen, right? That this G protein, this RAS protein is activated, right? In this transduction pathway, this response happens where a protein that stimulates the cell cycle is made. So this would only happen if this growth factor binds and it gets the signal that the organism needs to make more cells. What happens if this pathway is interfered with is then this is not happening correctly, right? We don't have this growth factor binding. This has just kind of gone rogue. And now this RAS protein is active with or without. So that can happen with mutations in the P53 gene as well. So if this goes wrong, there's no breaks. So you can see here in this cell signaling pathway, we have, instead of having this protein that inhibits the cell cycle being made here, that inhibitory protein is absent. So this, then there's no breaks on the cell cycle. It just keeps making more and more and more cells. So no breaks. Those are the main ways. So that's the main way that those mutations and those specific genes can interfere with the cell signaling pathway that then can lead to cancer. Okay, describe the process of creating induced pluripotent cells. Okay, so let me see. I'm wondering if I have that in here. I'll take a look. Because I saw that. No. That is not in here. So basically what they do is they will shock a cell And I'm looking to see if I have that even in here. They'll shock a cell and it kind of then reprograms into the embryonic stem cell. I'll make sure that you don't have to know. I think I wrote that wrong on the study guide. I don't think you necessarily have to describe the process of creating them. Just understand that they are adult stem cells that are then reprogrammed to act like embryonic stem cells. So I'm pretty sure I will look up and send a reminder if there's anything additional that you need to know about that. Other questions from chapter 16? Is everybody okay on the difference between, that's where I was going to go before, but I wanted to make sure. I don't know if I necessarily have any, I don't think I have any slides in here, but I know we had two different groups doing, everybody's okay on the difference between therapeutic cloning and reproductive cloning. Oh, look at that, Nicole. You read my mind. So therapeutic and reproductive cloning both use the exact same methods. So the method shown here is still going to be used where there's I guess I should say the beginning of the process. So we still have a cultured cell from an organism that cell cycle is arrested. And then we also have an egg cell donor that the nucleus is removed, and then these two cells are fused together, and then it's grown in culture. So where the therapeutic stem cells would stop is just it would be grown in culture. Right? Reproductive cloning is where then we would implant it, and then it would become another organism. So some people feel like therapeutic cloning is more ethical because it does not fully develop. We're not creating a whole nother organism. We're using those cells for research and for developing medicines and potentially to cure paralysis and regenerate heart cells and things like that. So same exact process, just there's kind of two different end goals there. Okay, chapter 5, information that we need to know. That was a long time ago. Sure. So basically what I just showed you guys on, I don't have chapter 5 pulled up, but basically just knowing this cell signaling pathway again. Here we have this right here, right? We have the ligand binding, reception is occurring, right? That's the first step. We have a transduction pathway. Sometimes this amplifies the signal. This, I know it's kind of a weird diagram. This would be the cell, right? So typically we have a intramembrane receptor because a lot of times these signals or these ligands are proteins. They can't pass through the cell membrane. So reception, transduction, some sort of response, which typically happens in the nucleus where DNA is being transcribed and then translated and a protein is made. This is the typical cell signaling pathway. That's why we're talking about it again here, is because this can go wrong and then that really affects the cell cycle. So just remember too, instead of having this intramembrane receptor, we could have an intracellular receptor. I don't have a picture of that up. But if there was a lipid instead, a lipid ligand or signal molecule, then that lipid could pass right through the cell membrane, bind to something inside of the cell, an intracellular receptor, and the process would just continue on the same way. So again, just remembering those 3 steps: reception, transduction, response, what that looks like, and then especially these diagrams of what can happen if it does go wrong is really what you need to understand with cell signaling for the test. Do we need to know the specific type of stem cells they use? I don't know what you mean. Can you clarify? Who uses? Nicole, that's your question. Like in a lab, what type of stem cells they use? I think, oh, okay. Sorry, I missed Jackie's question. Okay, so the therapeutic cloning uses the totipotent cells. Actually, they will use the pluripotent cells typically, so that they will develop into everything except placental cells. So yes, good question. So they will typically use, why can I not find that now? Here we go. They will typically use pluripotent cells. So cells will be pluripotent either if they're embryonic stem cells or if they're iPS cells. They're reprogrammed from just being an adult cell that's not a stem cell into, a lot of times they're skin cells, into a pluripotent cell. So good question. Thank you for clarifying. Anything else? We have about 4 minutes left. Did I miss anybody else's question? I don't think so. We're good? Okay, I'm not seeing any other questions. If you have anything else while I'm talking, put it up there and I'll answer it before I go. Again, I will look back on describe the process of creating iPS cells just to make sure. I'm really thinking I just wrote that wrong, but I'll look back and send you guys a remind if I do find something different that I need to clarify. Other than that, all right, well, I will see you guys tomorrow. Happy studying and enjoy the rest of your night. Bye. See if I can stop it.